Yes, good morning, all. Today we'll see the some important questions which can be expected in the examination. We'll see the important questions from all the means topics here. So first we'll see in the block one, the drug discovery, design, and development. So in this unit one, so the expected questions means for the short answer type. So we have two or three expected questions. Okay. So first, the stages. Okay, the stages of drug discovery. Okay. So the stages of drug discovery. So we know that what are the stages involved in the drug discovery. Okay. So what are the steps involved in the drug discovery? So you can give the stages involved in the drug discovery. So what is the step one? So in the sequence manner, you have to give the stages. Okay, so in this particular drug discovery, we have various stages. First, we'll see identification of lead for possible drug molecule. Right. So the very first stage for this drug discovery is okay. So first you have to identify the lead. Okay, lead compound means in which for which disease you are going to design a drug here. Okay, so for that, first you have to choose that lead compound. Lead means the start. Yes, lead means the starting material which is having the pharmacological activity or the biological activity. Okay, so the starting material for that, and next the synthesis of new pharmacologically active compound. Right. So then we are going to first you identify the lead compound, right? So then we have to synthesize that particular lead compound. Then studying the biological activities. Okay, so after synthesizing that compound, okay, first you identify the lead compound, then you synthesize that lead compound here. Then you identify the biological activity of that particular compound. Means whether it is satisfying the drug properties. So what are the drug properties here? So generally we have four drug properties here, like ADME properties. Okay, so drug property, nothing but the ADME properties. So you know that ADME, these are the five, okay, drug properties: absorption, distribution, metabolism, excretion, and toxicity. Right. So these are the drug properties here. So after stage two, you have to check that whether it is showing the biological activity or not, or whether it is satisfying all these properties or not. Okay. Then studying and understanding their interaction with different kinds of receptors. Right. So then you see the whether it is properly binding with your receptors or not. Okay, so we know that we have various receptors like proteins, enzymes, DNA, RNA, all these things. Okay. So, and ion channels also, these are the receptors here. So, whether it is properly binding with that one or not. So, you have to check that. Then, determination of drug absorption studies, distribution studies here. So, like ADME property, nothing but it is ADME properties here. And metabolic properties and toxicity properties. So, all these are the properties you have to properly check for this lead compound. Right. So, this is the stages involved in the drug discovery. Okay, so this is sometimes they will ask for the short answer here. And next to the expected question is what is it? Whole chloride drugs here. Okay. So drug. Hmm. Yes. So drug discovery without lead compound. Okay, so drug discovery without lead compound. So generally we call these are the serendipity of drug here. Okay. Serendipity of drug discovery. Okay. So serendipity of drug discovery. So this is also one of the important question which can be expected for the short answer here. So, sir, what is the serendipity of drugs, right? So generally the serendipity of drug means unexpectedly invention of the drug. Serendipity of drug, nothing but the unexpectedly invention of drug, right? So sometimes generally we are not took a project or we are not took a work for the invention of that particular drug here, but accidentally, okay, accidentally will invent a drug compound here. So that is called the serendipity of drug here. Invention of drug unexpectedly. Okay, so in this serendipity of drug, we have the discovery of penicillin. You have to explain the how the penicillin is discovered, how the penicillin is invented. So that example you have to give. Okay, so you write the definition of this serendipity of drug here. So what is the definition here? Unexpectedly invention of drug is called the serendipity of drug. Okay, or accidentally invention of drug is called the serendipity of drug here. And example for this serendipity of drug is in the discovery of the penicillin. So how the penicillin is invented. So you give the story of this penicillin discovery, right? So how it is discovered generally. So when that particular the scientist Alexander Fleming, right? So Alexander Fleming, generally he is a means a microbiologist here. Okay, Alexander Fleming is a microbiologist, and he was doing its work on the means he is took taken up a project of development of some bacteria here. Okay, some bacteria culture. For that he was developed the bacteria culture. Means he okay means he was taken the project of that bacterial development, and he was done all the means installation of all these things here, right? Right, and then and he forgot about that particular work here, and after sometimes he was observed that okay, so after sometimes, and he was busy with some other work, and after some days or for some days here he was observed when he was taken that petri dish or that kind of okay cultural dish, right? He found that he found the development of the bacteria, right? So here, for example, just to me, this is the petri dish. Okay, this is a dish here. Okay, where it is taken for this bacterial culture, right? And he found some formation of some fungus here, right? So this is a fungus. He found some formation of some fungus, and along with this, he found some development of the bacteria. Here is a bacteria, right? And here there is no bacteria. Okay, so he found that the bacteria is not developed. Bacteria are not developed where the fungus is formed. Okay, then he thought that there is a some means that fungus is having antibacterial activity. Fungus is having antibacterial activity, and means that fungus is preventing from the formation of the bacteria, or it is killing the formation of bacteria. Okay, and then later it is found that the fungus is right, Penicillium notatum here. That fungus is called the Penicillium notatum here, Penicillium notatum, and that Penicillium notatum is producing the penicillin. It is having the penicillin antibacterial activity, means that compound here, right? So that generally he was not he was not intention to discover the penicillin here, but means unexpectedly or the accidentally he was invented the drug here, right? So that is called the serendipity of drug here. Okay, that is also one of the, and discovery of librum also, that is also one of the example here, right? So you can explain that anyone, right? So you can explain the uh, penicillin, right? So penicillin discovery or the that particular Librum discovery. So you can explain anything, right? So first you give the definition, later you explain the story of the invention of that penicillin or your Librum. Okay. Then one more question can be expected from this one is what is it? Yeah. So random screening and non-random screening. Very short definitions here. Random screening and non-random screening. So generally the random screening and non-random screening. So random screening nothing but where that the invention of drug. Okay. So involves the invention of drug involves the it's generally okay. So when we are going to invent a drug, so the invention of drug is it is very tedious process here and it is very lengthy process here, right? So if you want to discover a drug or if you want to synthesize a drug compound here, so generally it takes 15 to 20 years time here. Okay, so it is very lengthy procedure. So that's why so we'll choose that screening process here. Okay, screening of molecules. Generally, what are the means the available compounds, right? In synthetic banks, okay, in the synthetic banks, so many number of millions of compounds are available here. So you can take all the compounds and you go with the screening of those compounds here, and you find out the lead compound and take that lead compound and you go for that modification of structural modification of that compound here, right? Then you will find some active compound from that. Okay, means that is trial and hit error, means trial and hit method only, right? So by error, trial and error method, hit and trial are the trial and error method. So you can find out that. So if okay, so if you find any drug compound, right? So it's okay if it is not if you are not unable to find that, means if you are not get any success hits here, then you reject that batch. Okay, you reject that group or the batch here. Then you go with some other group of compounds here, right? So that in this way, you choose a group of compounds and you go for that invention of that, means finding of that suitable lead compound, right? So that is called the random screen, right? So random screening means where it involves okay, where it involves that means there is no particular right the structure of that compound here. So we are not taking a specific what is we call that core structure here, right? So we are not taking or the choosing a specific core structure, irrespective of the structure, irrespective of the core, irrespective of the group, or irrespective of that ring size, whatever that, right? So we'll select all the compounds and we'll keep for screening here. That is a random screen. Okay. From that, uh, means by chance we'll get any success hit. And the non-random screening here, it is some systematic procedure, right? So non-random screening, it involves some systematic procedure where we first will, right? So we'll choose the lead compound which is having pharmacological activity. Okay. So to improve the activity, we'll go for this non-random screening only. Okay. So that is the difference of this. It's one of the important questions which can be expected for these short answers here. Then one more important question is very frequently asked question is the medical folklore. Okay, folklore drugs here. So folklore drugs very important here. This folklore drug is very important. So you can right. So folklore drugs. So folklore drug nothing but where yeah all these are screening. So for the screening whatever what are the types of compounds you will choose, right? So various sources for these compounds here. There is the purpose, enum syntaxons, marine sources. Yes, this is the right. So medical folklores here. So this is important, very important question. So what is medical folklore? So folklore medicine, nothing but the ancient, right? In the ancient days, there is no development of much development of the technology here. Okay, so generally the peoples, right? So the peoples, they are curing their diseases, the wounds, okay, diseases, wounds, or anything, right? So any what is that, abnormal conditions, they are curing by taking the medicinal plants here, right? Means the plant, what is that, leaves are, okay, fruits are, leaves are, and roots, whatever. But generally, they don't know the importance of that plants here. They don't know the importance of that leaves. Okay, they don't know the importance, medicinal importance of the particular plants or the trees here, right? So unknowingly, they are taking that. Okay, right? So that is called the folklore drugs here. Other folklore, medical folklore, right? So for the okay, you know that variety of, yeah, right? So variety of that, what is that? Variety of drugs are invented from the plants here, right? Natural products like your anti-malarial quinine, okay? So and morphine, quinine. So all these are the medicinal plants here, which is having the uh, pharmacological activities, right? So folklore, nothing drugs, nothing but where the right. So the obtained from the plants, right, and from forest here. Here there's some examples: reserpine, morphine. Okay, so all these are the examples for this, and all these are okay. So from unit one, so you can expect all these things here. Then we'll go for the unit two. Next, design of agonist and antagonist, development of capability. Right. So in this particular topic, right, so in this, right, so agonist and antagonist. From this, so you can expect, means they will ask the definition of agonist and antagonist, right? So they will ask what is agonist and what is antagonist here, right? So agonist, nothing but where, okay, that where the drug compound, right? So where the drug compound are the natural neurotransmitter. Okay. So when the concentration of the, right, when the concentration of the natural neurotransmitter or the chemical messenger, right, is low, right? So then to increase the concentration of that natural neurotransmitter, the chemical messenger, right? So we use the drug compound. So that is called the agonist here. Means that agonist which is increasing the concentration of the chemical messenger. That's all. Right? How do you define here agonist? The medicine or the drug, okay, the drug compound or the medicine which is increasing the concentration of the natural neurotransmitter or chemical messenger is called the agonist here. And antagonist. So what is antagonist here? So when the concentration of the chemical messenger is high, okay, so to lower that concentration of the chemical messenger is called the antagonist here, right? So like captopril, you can see that, okay, captopril here. So captopril, so generally it lowers the blood pressure, okay, sorry. right? So it lowers the blood pressure. Captopril lowers the blood pressure. So when the blood pressure are the right, so when that particular AC, right, AC, angiotensin converting enzyme, it is producing the excess of angiotensin 2. Okay, so where it overstimulate the smooth muscles around that heart. Okay, so then it cause for the more development of the blood pressure here, right? So in that case, when you use a captopril, enalapril, okay, captopril, enalapril here, ramipril. So all these are the drugs. So which inhibit the AC activity. They inhibit the angiotensin converting enzyme. They will bind with the angiotensin converting enzyme and they will uh, uh, prevent from the activation of the angiotensin chemical messenger here. Then automatically the blood pressure comes in the normal mode, right? So this is a acting as an antagonist here, right? So that is a definition of agonist and antagonist. Then in this particular topic, one more expected question is development of salbutamol here. Very important, right? Development of salbutamol. So how the salbutamol is developed, right? So you know that the salbutamol, it is right. So it is a uh, anti-hypertensive drug, right? It is anti-hypertensive drug and anti-asthmatic drug, right? It is acting as, it is multiple functions, multiple pharmacological activity. So it is acting as a uh, anti-asthmatic drug and it is acting as a anti-hypertensive drug and also it is acting as a what is that? Means it is used during that what is a delivery, right? So it is used for the delaying the premature labor. Okay, so these are the uses of this salbutamol. And actually this salbutamol development, right? So development salbutamol, it is an anti-hypertensive drug or the asthmatic drug. The development of this salbutamol is taken by adrenaline hormone, right? Adrenaline hormone as a lead compound. Adrenaline hormone itself, right? Adrenaline hormone itself is taken as a lead compound here. Okay, means this is comes under the drug discovery with lead compound. Okay, drug discovery with lead compound. So, like in the topic one, unit one, we have seen that the drug discovery with lead compound and the drug discovery without lead compound, right? So with lead and without lead. So with lead compound means where the lead compound is chosen for this development of the synthesis of the drug compound. Without lead means there is no lead compound for the synthesis here directly, right? So the whatever the lead or whatever the compound is invented itself is a drug compound. There is no further development of that. Okay, so here the salbutamol, it is taken means it is developed by taking adrenaline as a lead compound here. Okay, so adrenaline, you know that it is a hormone. Okay, adrenaline hormone. So and how this adrenaline hormone is developed as salbutamol, you can see. So here generally this okay, the adrenaline. Okay, so adrenaline, it is beta, right? So it is beta 2 receptor active. Okay, it is adrenergic, beta 2 adrenergic receptor active active chemical messenger generally, right? But it is having the short duration of action, right? It is having the short duration of action. That's why they could gone with the modification of that the structure of that particular adrenaline. Okay, and for that, what are the means what are the steps they involved, how they finally developed the solvent. You can see. Yes, you see. Yes, the adrenaline, it has a short duration of action here, right? Adrenaline itself is means generally adrenaline also having the it's adrenaline is having the activity, but generally it is having short duration of action here. So for that, they are developed some means they are done some modifications, and what are the modifications they will done? So you can see here, yes, and some other uh, disadvantage of this adrenaline is so it is not selective only to beta 2 receptor, right? So it is wide range of this selectivity. Means it is selective towards the beta 1, beta 2, alpha, right? So alpha 1, alpha 2, and gamma. Generally, the adrenergic receptors, right? So yeah, the adrenergic receptors having five types here, right? Alpha 1, alpha 2, right? There are five types here. So here is alpha 1, alpha 2, beta 1, beta 2, and gamma. So these are the five types of adrenergic receptors here. So your adrenaline chemical messenger, it is selective towards all these receptors here, right? So, but only beta 2 receptor is activating the smooth muscle. Okay. So we need only exclusively beta 2 receptor, beta 2 selective receptors. That's why we are gone with some modifications of this adrenaline structure here, right? So first, they are done the replacement of this N-CH3 group, right? So N-methyl group replacement of N-methyl group is with isopropyl group. Okay, so they are done the modification by removing the methyl. They are introduced the isopropyl group. Then it becomes isoprenyl, right? So this adrenaline becomes isoprenyl here. Then this isoprenyl, it is selective to beta receptor. Right? So the generally when this NCS is replaced with the isopropyl group, they found that it is selective to beta receptor. Right? So but we need beta means again there are two types, beta 1, beta 2 here, right? So we need only beta 2 selective, not beta 1 here. But it is inversely beta receptor selective compound. Right? So when this adrenaline when this methyl group in the adrenaline, N-methyl group in the adrenaline, when it is replaced with isopropyl group, so it becomes beta selective here. So in the beta, we have beta 1, beta 2, but we need only beta 2 selective compound. That's why this, right, so this is rejected. Again, they introduce some other groups here. They replace some other group. And one more difficulty with this isoprenyl compound is one of this OH group in the catechol, right? So this one of OH is, it is converting into, right? So it is methylating. It is methylating with COMT, right? So catechol O methyl transferase enzyme. Catechol O methyl transferase enzyme. So with this enzyme, this particular one of the OH group is converting COMT here, right? So catechol methyl transferase enzyme. So it is converting into methoxyl group here. The OH converting into methoxyl group. So this is also one of the interference in this, right? So this is also this modification is also not work out. Then they replaced this OH group, right? They converted this OH group uh, into carboxylic acid, COH, right? So again, they replace the OH group as a COH group. Then what happens here when this OH group is, right? So OH is replaced with the COH group, then the entire structure becomes inactive. The entire structure becomes inactive. So this modification also not means giving the good result, right? So then they are gone with the further modification with ester and amide. Okay, ester and amide. Then what happens here when this ester and amide conversion is done, then it is acting as an antagonist, right? So then this modification is giving the means what is that good result, right? So when this OH is replaced with the ester and amide group, then it is acting as an antagonist. So we need this property here. Okay, but what happens here? These right. So these compounds is having the long duration of action here. Long duration of action, even though it is used compound, right? Because there is no further modification. They didn't went for the further modification, and half an hour. Yeah. So it is introduced in the market anti-hypertensive drug and anti-asthmatic drug. But later it is developed uh, by modifying with means this ester and amide groups are changed as a right. It is changed as a CS2OH group, right? Then it is giving the and some sulfonamide group and uh, what is that methyl alcohol group here. So then these are having the 100% results, right? It is having the good results. Okay, so this is the development of salbutamol, and one more is development of what is that captopril also, right? So development of captopril. So in this captopril development, right? So here also generally this captopril it is anti-hypertensive. Again, this is also anti-hypertensive, right? And generally it inhibits the activity of AC, angiotensin converting enzyme, right? So you know that AC it is converts the angiotensin 1, right? So angiotensin 1 is converting into angiotensin 2 in the presence of AC, right? So it is involving in the hydrolysis of the polypeptide here because it is angiotensin, it is a decapeptide. It is a decapeptide here, and it is hydrolyzing two peptide bonds here. It is cleaving the two peptide bonds, and it is converting into octapeptide. Angiotensin 2, it is octapeptide here. Okay. Right. So, but when this ASE failing its function, when the angiotensin converting enzyme, it is overactivated or the overstimulated, then the more conversion of angiotensin 2 is takes place, means where the angiotensin 2 concentration becomes high, right? So where high production of the angiotensin 2 is takes place, right? So then what happens here? It overactivates your smooth muscles around the heart, and it causes for the more blood flow in the blood vessels here, right? So then it is causing for the development of high blood pressure, high BP. Okay, so if you right, so if you inhibit the function of this ACE, then the concentration of angiotensin 2 will be decreases here, right? So for that, the captopril is developed here, where the captopril is inhibiting the function of ACE, right? So for the development of captopril, where they are right. So because they are taken the example of the carboxypeptidase here, right? So carboxypeptidase, generally this carboxypeptidase enzyme, it is also involving in the hydrolysis of the peptides here. It is also involved in the hydrolysis of peptide. Okay. So both means here the ACE and carboxypeptidase, right? ACE and carboxypeptidase both are having the same function. Okay, same function here, same function. So they are taking the inbuilt structure of the carboxypeptidase as a lead compound, right? In the carboxypeptidase, the inbuilt compound, what is that inbuilt compound in this carboxypeptidase? One second, it is yes, N-benzyl succinic acid, right? So N-benzyl succinic acid is the inbuilt structure in the carboxypeptidase here. So this N-benzyl succinic acid is taken as a lead compound, right? So this is taken as the lead compound for the development of what is that? Captopril here. By some modifications, they are achieved they are succeed in the invention of your captopril, right? So, and what are the modifications they are done here actually here? So where they are this general the benzyl succinic acid having a 6-membered benzyl side chain, but they are introducing right proline here, right? So in place of benzyl group, they are they are modified as a proline, and the side chain is same as your N-benzyl succinic acid here. Okay. Then they developed this one. Okay, after modifications, they developed this structure here. And the difficulty with this structure is, right? So generally, this carboxypeptidase, right? Carboxypeptidase enzyme, sorry, AC. Okay, so AC enzyme, right? So it is having the interacting point with this drug, right? So where this, right? So where this is the peptide structure here, and you see this acid COH group is, so it is forming the ionic interaction with this arginine unit, okay, and where this zinc metal also there. So this is also able to form the ionic interaction, and where this the S1 and S2 pockets are left here, right? S1 and S2 pockets are left. So in the means in the active site of your AC, the active site of AC. So where these packets are unoccupied. So due to this unoccupying of these pockets, S1, S2 pockets here, where the proper fitting is not done, where the proper interaction or the proper fitting is not done, then where the activity is low. That's why they are introduce the methyl on the right. So the methyl on this particular. So, yes, you see, right. So here the okay. So this COH group, it is interacting with your okay arginine unit, and this okay. So it is interacting with zinc metal, and this CO group is it is occupying this S1 pocket here, and S2 pocket is left here. It is unoccupied. That's why here they are introduced the methyl group on this CS2 group. Right? So if you introduce a methyl here, then this CSC group is occupying this S2 pocket. Then you can see the proper fitting or the proper binding of your angiotensin converting enzyme active site here. Then the more activity will be found, right? So in this way, they are modified this particular structure, and later it is right. So in place of OH, so they are replaced with the thioalcohol group here. Here is a right. Here is oxygen, not here methyl, and finally they are developed this captopril structure, right? So where the N-benzyl succinic acid is taken as a lead compound with reference to this, okay, with reference to the carboxypeptidase enzyme, right? So by seeing the carboxy peptidase enzyme, right? So they are uh, right. So they means they are taken the reference this carboxypeptide and enzyme for this AC. Then the inbuilt compound N-benzyl succinic acid is taken for the taken itself, right? So taken it as a lead compound and they further developed the captopril compound or the drug structure. Okay, so that is the uh, means the expected questions from this one. Um, yes, yeah, studies here. Yes. So in the SR studies also same thing. Yeah. So in the SR studies, yeah. So in the SR studies, SR nothing but structural activity relationship. So in the SR studies, we have these modifications here, right? So these are the some modifications like variation of the substituents means these are the sections where you have to do the modifications here for the development of the efficient drug. Variation of the substituent, extension of the structure, extension of the chain contraction, and ring expansion and ring contraction here, and ring variations. Uh, what is that? Isostats and bioisostats. This is very, very important. Okay, isostats and bioisostats. Then simplification of the structure, rigidification of the structure. So all these are the structural requirements, right? All these are the structural requirements in order to develop the uh, efficient or the what is that more active drug compound. Okay, the potent drug. So variation in the structure means you can do the modification of the structure here. Okay, so here is right. So alkyl substrates. You can replace the alkyl substrates with different functional groups like OH and NH2 or and NHR, so acids, all these things. Right? And you see the means binding with your receptor actives, how effectively it is binding. So you can see after replacement of the alkyl substituent with some functional groups like OH group, acid groups, and amide groups, some amine groups here you see the binding interaction with your receptor active site here. How effectively it is binding with, then generally the more effective binding gives a more activity here, right? So you see that changes of that modification and extension of the structure. So you can extend the structure here, right? So you see, for example, so here this is the okay. So this is your morphine structure, okay, and hypnotic drug. So this morphine structure where the N, right? So here is a NCSC group is there, NCSC group here, right? So this is the heterocyclic ring. So this CSC group is replaced with the benzyl group, right? Right? So this CSC group will replaced with the benzyl group. Then the extension of structure. So where it calls for some rigidification of the structure, and it sometimes it is causing for the increase of the activity. Okay. So you see by changing the substituents, the side chain or whatever, right? Okay. And here is a SS enolopyl, right? So where this SS captopril, right? Here the captopril structure with further extension with some benzyl substituent here. So it is uh, forming as your, it is developed as your enolopy, right? So with the structural extension, the modification, in side chain modifications, they will develop this particular enolopyls. So with the structural extension, so we got the variety of analogs, drug analogs here. Okay. So all these are the some structural modification, and here the ring expansion and ring contraction. Okay. So ring expansion. So this is also one of the structural requirements for the getting the more efficient drug compound here. So ring expansion means we can change, you can increase the size of the ring here. For example, here you see, right? So here you can see this naphthalene ring structure here, the core structure, right? So the naphthalene core structure is there. So where this the okay, the A B C, okay, there are three-membered ring. So where this B ring is expand, right? The B ring expansion is takes place when the six-member becomes seven-member, right? So we are expanded the B ring here as seven-member. Six-member becomes seven-member. Then it may be going to increase the activity here. Okay, so means this is the only the for what is that? The suggestion here. Okay, so you do this modification and you check uh, whether it is going to increase the activity or the decreasing the activity. If it is going to increase the activity, then you can proceed with that modification. If it is decreasing the activity, then you uh, go with some other modification, right? So like that. So that is some. It is purely trial and error method only. Okay. So this is a ring expansion and contraction here. And one more important question can be expected is isostress. Very, very important. Okay. Isostress, nothing but generally changing of the group. Okay. Changing of the group with its iso electronic or the isovalency. Same valency. Okay. With the same valency or the same electrons. It, it would not have causing for the, right? It would not causing for any biological activity, changing in the biological activity, right? So the replacement of the isovalency group or the same valency group or the same electron group or the isoelectronic group. Isoelectronic group or the same valency group is not causing for change in the biological activity. That is a isosters. Okay, the definition of isostar nothing but okay, the changing of replacement of the one group with its same valency group or same electronic group is not causing for the change in the biological activity. It's called the isoster. For example, you see thio SS group and NH2 group and CSC group, right? So these are the same valence with your hydroxy group here, same valence with the hydroxy. So there is no change in the biological activity and no much change in the biological activity, right? So you can, in place of OH group, so you can take the SH group here, right? So like in the previous case, in the right. So we're in the captopril, right? So in the captopril, uh, in the captopril where this, okay, so it is replaced with the SH, right? So like that. So where this OH group and these are the uh, isoster. These are the isosters. So you can take OH or the SH, right? Yeah, the bioisosters. Bioisosters are nothing but where the groups are having the biological activity and replacement of the uh, the replacement of that particular one group with its right which group which is having the equal valency, same valency group or the same electronic group, isoelectronic group, is not causing for much change in the biological activity, right? That is called in bioisosters. Okay, so these are the some important it's isosters and bioisosters and rigidification or the contraction of the sorry, expansion of the ring size or the contraction of the ring size. So these are the important things, expected questions from this. And then the next is six. Yes. So next the combinatorial synthesis. So here is a combinatorial synthesis. So generally, what is combinatorial synthesis here? The definition. Generally, this definition of combinatorial synthesis, production of the large number of compounds in a short period is called the combinatorial synthesis. The combinatorial synthesis, we have uh, many examples, right? So important questions from this. So here is a need of combinatorial synthesis. It's important. And resins, linkers, okay, resins, linkers, need of combinatorial synthesis, Webster, right? Hot and tea bag method. Here, what is it? Hookah method, right? So these are the important, right? Okay, so first, need of combinatorial synthesis. So what is combinatorial synthesis generally? Production of large number of compounds in a short period is called the combinatorial synthesis. So what is the use or the need of this combinatorial synthesis? So for this category, generally you can see need of combinatorial synthesis. Okay. So here we have three set of, okay. So we have three building blocks here, right? So three set of building blocks, right? Where A1, A2, A3, B1, B2, B3, C1, C2, C3, right? So we have three set of building blocks. So with help of these three set of building blocks, we can produce the 27 products here. 27 products, right? We have only three set of A1, A2, B1, A1, A2, A3, B1, B2, B3, C1, C2, C3, right? So we are taking these only three set of compounds here, only three set of compounds. But how many compounds we are getting? 27, right? So you can see that is in a short period, in a short period with by using the very few starting material, we are getting large number of products here. That is the advantage or the use of or the need of your combinatorial synthesis. Right? How we are getting, you can see. For example, A1. Okay. So first you can combine with the B1 here. The A set of A type of building block. Generally, this combinatorial synthesis, right? Combinatorial synthesis, generally it is applied in for the peptide synthesis and in the field of peptide synthesis and drug discovery, right? So drug discovery here. So you see here first we are getting this A1 B1 here and A1 B2, A1 B3. Again we are taking the B type of here A1 B1 C1 here, A1 B1 C2, right? And A1 B2 C2, so like this. So total there are 29 compounds are produced in a single step here, right? So in a single step we are getting 27 of compound. By using the 3 building blocks, we are getting 27 compound here. So by keeping only one reaction, we are getting 27 products here. If you go with the conventional method, or the traditional method, so if you keep one reaction, you will get one product, right? So here in the combinatorial approach, so instead of keeping the 27 reactions, we are right keeping only one reaction, we are getting 27 products. If you go with the conventional method, you need to keep the 27 reactions to get the 27 products. But this approach is useful, right, than your conventional method. That is the use, the advantage, or the need of your combinatorial synthesis. And yeah, the requirements, right? For this combinatorial synthesis, some requirements are there. So what are the requirements here? So first, here is a cross-linked insoluble polymer, right? So resin beads are very important here, right? The basic requirement for this combinatorial synthesis is the resin bead here. Okay, the solid support. Resin bead nothing but it is a solid support, and the anchor or the linker is a right. The linker here, the linker is the next requirement for this combinatorial synthesis, right? And what is that? A suitable, right? A suitable protecting group also, right? A suitable protecting group is also one of the important requirements, and suitable conditions also. Okay, suitable conditions is another requirement here, and protecting and deprotecting groups also required for this combinatorial synthesis. And the solid supports are the resin beads here. The solid support are the resin beads, so very important here. So Merrifield resin. So Merrifield resin is important, very, very important. Right? So Merrifield resin is very, very important. So generally Merrifield resin, it is a poly. Okay. So the polymeric structure of the cross linkage of your styrene with divinyl benzene. Okay. So here is the right. So it is a styrene. Sorry. Right. Here is a divinyl benzene where it is cross linked with your styrene. This is your styrene. Right. So again, one more styrene is there. Okay. Right. So it is cross linked. Right. And one more styrene, it is cross linked. So like that. And near below also same thing. So by this cross linkage of the styrene with the okay, it is cross linkage of the styrene with the divinyl benzene, and it forms a manifold resin. So you can see this structure, how this manifold resin is formed. And sapphire polymer resin and right, so tentagel resin. So all these are resin examples for this. And the linkers. So linker, generally it is active functional group here. It is active functional group. It is attached to that particular what is that solid support. Okay. And one more thing is methods of parallel synthesis. Yes, here is the important one is a Hoopston tea bag method. Right. So this is also Hoopston tea bag method. So it is having the procedure you can see. Right. So like the Hoopston tea bag method. So it is polypropylene mesh. Polypropylene mesh is there, and it is closed with your polyethylene glass here, right? And it is having some, right? So all these are acting as a chambers here for the means reaction vessel, and where the reactions takes place in this tea bags. We call these are the tea bags here, and it is labeled. Already each tea bag is labeled. Okay, so this is, and this one. This is also important. Furka method, mix and split method, right? Furka method. So here the stages, first the attachment of your building block with your resin bead, then the mixing here. Okay, you attach and you mix, then you add another building block and you make that reaction and you do the deep protection and you get the products. This is the mix and split method. Okay, so this is Furka method. And so next is QSR, qualitative structure activity, unit 7. So in the QSR, here also we have some important results. So here the top steric factor is important here. So top steric factor generally in the QSR, right? So QSR, qualitative structure activity relationship. So it deals with the physicochemical, means the relation between the biological activity with the physicochemical parameters, right? So in the physicochemical parameter, we have various physicochemical parameters like partition coefficient, okay, log p value, partition coefficient, and here the top steric factors here, okay, and electronic factors. So all these are the factors which is yeah, affecting that biological activity, right? And in the top steric factor explains the right explains the presence of, right, increase in the bulkiness, steric nothing but the bulkiness here. So the bulkiness, how the bulkiness is affecting the biological activity, it is explained by taking some hydrolysis of this ester here. Okay, so where this particular ester compound, right, so this is the ester, when it is undergoing the hydrolyze with your hydrolyze, and it is converting into your carboxylic acid. Okay, carboxylic acid and alcohol here. So where this involves a nucleophilic substitution here, nucleophilic substitution, and this rate of hydrolysis, right? Rate of hydrolysis is affecting with your the group size or the substituent size here, right? So for example, this is R, where the very small size alkyl substitute. Then when this is the R1 here, the R2, just you say this is R3 here, right? So with increase of the size, right? So with increase of the substituent size, okay, so it is causing for the decrease in the activity or the what is that, rate of hydrolysis is going to decrease here, right? Here you see from you see the increase in the R group here, right? Here is a decrease in the K value. K nothing but your rate constant, right? So in this way, it is affecting your biological activity, right? So it is explaining the difference or the effect of your substituents or the bulkiness with your biological activity. It's things plot. Yeah, yeah, the Hans analysis are also one of the factors here. So Hans analysis. So what is Hans analysis? The Hans, he was derived the relationship for the all the parameters, all the physicochemical parameters, right? So he was taken up the all the parameters like okay, partition coefficient here, and the electronic factors or the electronic parameter, and steric parameter, right? So all the parameters or the physicochemical parameters are taken up for to build a correlation graph here, right? And he was derived the relation how it is affecting the biological activity, one log 1/c, right? So generally in the previous where we have seen the individual parameters, but here all the parameters are taken up to build a equation here, right? So this is the what is it, Hans analysis. And the Hans analysis, the disadvantage, it is having some merits and demerits also. And what are the merits and what are the demerits generally? You can see here is a okay. So the demerits are the disadvantages are the demerits are the okay demerits of your Hans analysis. Where the conformation changes at the receptor is ignored and metabolism does not interfere here, and linear free energy terms relevant to receptor affinity are additive here. Okay, and the polarity lipophilicity relationship is linear or the parabolic, right? So here the conformation change at the receptor is ignored. So generally, this particular uh, right, so the conformation changes in the receptor. If you assume this is your receptor here, right? So if any compound, right? So here is the compound, for example, a drug compound, right? So when this drug compound is occupying this receptor active site, this drug must be matching with the conformation of your receptor active site. Then only the proper adjustment or the proper fitting or the proper binding of the drug is takes place, right? That is generally to explain the drug receptor conformation is we have various theories here like lock and key mechanism and induced fit mechanism, okay, and 3-point contact model here. So lock and key mechanism, it explains that when the geometry or the shape of the drug compound or the receptor, the drug compound or the ligand is matched with your receptor geometry, then the proper fitting is possible. Otherwise, there is no
binding of this drug or the ligand with your receptor active site. That is explained by the lock and key mechanism. Later, it is found that the induced fit mechanism. So where the means as per the lock and key mechanism, there is no conformation changes which takes place here, right? There must be a proper matching of the geometry of your receptor means drug and receptor. But in the induced fit mechanism, generally there is no condition, there is no required, right? So there is no condition to have the means the matching property here, right? So the matching is not the criteria for this binding of the ligand with your receptor, right? So where the lock and key mechanism, as per lock and key mechanism, the uh, right the matching of geometry, same geometry is required, but as per the induced fit mechanism, the same geometry is not required here. Okay. So as per this induced fit mechanism, generally when that receptor, when this ligand or the drug compound is approaching the receptor active site, so your that particular receptor is converting the conformation according to the geometry of your ligand or the drug compound. Means there is a conformation changes, right? Conformation changes is takes place. Then it is facilitating for the binding of your drug compound with the receptor, right? So, but as per this induced mechanism, uh, the conformation changes will be takes place. But in this Hans analysis, he was ignored the right. So where the conformation changes is ignored in the receptor, right? So that is also one of the mistake or the disadvantage of this. And the advantage of this Hans analysis is so all the descriptors are taken to build a right to build an equation here, and predictions are quantitative here, where the statistically it is quantitative, and quick and easy conclusions are done, right? And where the potential extrapolation substance is not included in the original analysis, right? Next, Craig's plot. So this is also one of the important, right? So Craig plot. So generally, the to calculate the right the log 1 by c value, that is a biological activity. So all the groups or the compounds are divided into four coordinates here, right? Four coordinates, and where these coordinates are having the positive values and the negative values here. So you can take your required, okay, you can take your required values to get the better active, means to get the better activity, right? So for example, if you take the plus alpha and plus pi value here, sorry, plus sigma and plus pi value, so you will get the more biological activity here. It is causing for the poor biological activity. So you can choose from that where you get where you have these positive values here. Generally, yes, in the right coordinate, right? So in the positive lobe, okay. So in the positive lobe, in the right side, you have the plus sigma value and plus pi values. So you can choose the groups which are false in this coordinate, right? Then it is easy to find out that substance which is having the plus values, plus sigma values, plus pi values here. Okay. Or if you want one plus value and one negative values here, so you can choose from this left side here. So like that. That is a means main advantage of this Craig's plot. So you can directly pick up the group which is causing for the activity. Right? So that is and the top seal decision tree. This is also one of the important question which can be expected here. So top seal decision tree is it is very easy to uh, interpret the biological activity. Means here to take the decision which group is included to increase the activity. Okay, so by generally this topsil decision tree, so it involves by trial and error method only. Okay, so you see, for example, here in this we have three decision points here, two possibilities, right? So when you introduce one group, okay, so then we have three possibilities here, right? Where For example, here you take some example. We are taking with, for example, this is a benzene here, right? So we are taking benzene as a lead compound, and we are making this. We are replacing this hydrogen with chlorine, for example, right? Right. So here is just to assume this is some X is there. Then we have this is 1, 2, 3, 4, 5, 6 here. Okay, so these are numbering we are given, and now you see, right? So with the two, at second question we are introduce a chlorine. Okay. Or at fourth question, for example, right? So as per this flow chart, where this right? So where this four, right? So where this is para. Okay, so at para position we are introduce a chlorine here. Right? So we are introduce a chlorine at para position. So then we have three options, means three possibilities here. So the introduction of this chlorine, where it is low value or equal value, or the sorry, this is medium. Low. Yes, right. So either low value. Here is a equal value. Here is a M, not E here. Okay. So M means more, higher value. Here is a low value. Here is a higher value. Here is a equal. Okay. So where L means low, here is a E. E means equal. Here is a M. M means more. Okay. So these are three possibilities when you are substituting one group or the atom with another group or the atom here. So we have three possibilities here. So it is causing for decreasing or equal or the more. Now from this, you reject this lower value, which is if it is causing for the decrease in the value, you can means you don't go with this one. If it is causing for the equal value, then we won't choose this route here. We won't choose this means substituent. If it is causing for more value here, then we go for this. Okay. So you see what is happening when you are introducing this substituent here. So what is happening, or whether it is causing for higher values, then you go take this chlorine at that particular fourth substituent here, right? So, for example, if it is not right for increasing the value here, then you go with the, for example, here you see, okay, here the low value. Then you reject this chlorine at fourth position. So instead of this chlorine, you go with some other group. We are introducing methoxyl group. Okay. So when you introduce the methoxyl group, again we have four options, right? Whether it is lowering value here, means less value, equal value, or the more value, right? So like that. So that is a cluster analysis. So next, drugs acting on metabolic acting on process. In the metabolic process, generally, yeah. So the sulfonamide drugs are important. Yeah. So in the sulfonamide, you see, so this is the sulfonamide drug. So with the and the SAR of sulfonamide. Right. So SAR means the structure activity relationship. So here you see various means modifications are done during that invention of sulfonamide. You see the exchange of sulfonamide group with this particular, so where it is retains the activity here, and the mono substitution at para amino means para amino group here, where it is increase the activity here, and dye substitution, it is decrease the activity. So all these are the it's various right the SAR studies steps here, and all these are analogs here, and mechanism of sulfonamide is important, right? Yeah. Mechanism of sulfonamide. Right. So here you see generally this sulfite is antibacterial drug. Okay, so where this so here THF is converting into DHF, DHFR. Okay, yeah, DHFR, it is converting into DHFR. Okay, and this is DHF, it is involving in the, right, so it is involving in this. Right. So here THF is converting into this is tetrahydrofolic acid, and it is converting into thymidine. Thymidine, then it is forming the DNA. Right. So this is the route here, pathway. Okay. So your right. So your sulfonamide. Okay. The sulfonamide is having the similar structure with the PABA, para-amino benzoic acid. Right. Para-amino benzoic acid is, it is binding with your dihydrofolate, right? Dihydrofolate group. Dihydrofolate group. Dihydrofolate, right? And it is binding with this dihydrofolate, and it is converting into dihydrofolic acid. And dihydrofolic acid, further it is uh, combined with your this particular what is that? That dicarboxylic acid, and is converting into thymidine. Then further thymidine converting into DNA. So this is the normal pathway here. Okay? So when this right, so when your particular uh, sulfonamide drug is taken, right? So this sulfonamide drug, it is compete with your PABA, para-amino benzoic acid. So this PABA, okay. So instead of this PABA, your sulfonamide drug is binding with at this center here, right? And so generally this is not the structure of this one. Okay. So where the pathway is diverted here, you see, this is your dihydrofolate acid, but this is a false mechanism, right? Where this is the sulfonamide. Okay, this is PABA. Okay, so it is where your sulfonamide, it is compete with this para-amino benzoic acid, and where it is stopping this DNA formation of the bacteria. Okay, in this way, it is stopping the bacterial growth. Okay. So these are some important questions, and remaining. Okay, so already we discussed in the class some important questions. Okay, so I'm stopping here because already the time is qualified. Okay. So if any important questions are left, so I will give the important questions. I will share in your group. Okay. So I'm stopping here.
Okay, sir. Thank you, sir.